And here we are. It's 1 minute past the hour, so let's get started, everyone. Welcome back. This is David Regan, CEO of Sona Nanotech, and I have, of course, with me Dr. Carmen Giacomantonio, who is our Chief Medical Officer, and we're really excited to share with you an update. It's been some time since our last investor webinar. We love to have these opportunities to be able to answer questions, share new information with you, and we do have some new information today, both on further clinical tests. We promised to build a mountain of data, and that's exactly what we've done and continue to do. And it's all producing great information. We're keen to tell you a little bit more about it and some more color around it today. And then, of course, provide an update on where we're going next, specifically with our first in-human early feasibility study. But what increasingly we're finding, and what you're going to see on this cover page here, is, of course, targeted hyperthermia is in and of itself an immunotherapy. It's activating the immune system. That's increasingly what we're seeing, not just reducing the size of tumors. And so there's a new color we're putting over this, and you're seeing that we're priming the immune system to enable immunotherapy, but it is, in fact, an immunotherapy, albeit a device. So we're going to be making forward-looking statements today, so I would refer you to disclosure documents and the cautions therein of all eventualities, both expected and unexpected in the future, to anticipate. Agenda for today. So 3 areas we want to talk to. We're going to be very brief around description of targeted hyperthermia. If you're new to following the Sona story, and I know there are a bunch of people who are, we'd refer you back for further in-depth presentations in some of our past webinars. Second of all, we're going to go right to what is new since the last time we've spoken. And then finally, where are we going, and the updates around that. So as to Sona, what is Sona? What do we have? What is our therapy? Sona's core technology is a particular type of gold nanoparticle. There are many types of nanoparticles. We make one called a nanorod. The nanorods are, of course, highly functional because of their elongated nature. They have an aspect ratio, which means you can tune the length and width to match the energy waves from, in this case, a laser that we're using. Makes them highly functional. That's why we like them. The problem with gold nanorods typically is that a toxic substance is required to make them into that elongated shape. Our core technology is a patent that uniquely allows us to do that, to elongate them without the use of toxins. And that is critical if you're going to use these particles in vivo for nanomedical applications. And of course, that's what they're best suited for. We've been honored to work with the NCL in Fredericksburg, Maryland, which is set up, of course, by the FDA and the National Cancer Institute. They have validated our materials and have been wonderful partners and have sat on our side of the table in our meeting with the FDA. So good partners to have. So what to think about when you think about Sona is that we have uniquely biocompatible gold nanorods, and those are ideal for working in nanomedical spaces. So now, what's the problem we're trying to solve? I'm going to turn it over to Carmen, who's a surgical oncologist and can tell us a little bit more about the environment and what problem it is that we are seeking to solve.
Thank you, David. So what we're going to talk about really is the current state of the art of immunotherapy. So immunotherapy, and we're going to look at pembrolizumab as an example of a PD-1 inhibitor, programmed cell death receptor 1 inhibitor. So this just briefly review that this receptor is activated as soon as a T cell, the cytotoxic cell that kills cancers, is activated. It turns on a receptor so that it has a limited shelf life, so that it doesn't overreact to a situation. This is both a benefit and a curse. To overcome this checkpoint, PD-1 inhibitors and other immune checkpoint inhibitors have been developed, this being one of them. The discovery of this in the role of cancer was actually, related to a Nobel Prize. These checkpoint inhibitors, when turned on, will enable immune activity as long as the immune activity has already been activated. So what's necessary for a checkpoint inhibitor to work is that there is a cancer immune system relationship already at play. So what we've seen over the past number of years, and now these are more than 10 years in clinical trials and now in standard of care clinical practice, is that with a single agent, pembrolizumab, choose whatever PD-1 inhibitor you like, there are several on the market, the maximum response rate is about 35%. Now, there's varying reports on this, but essentially with a single agent, meaning that we give only pembrolizumab or a PD-1 inhibitor and we block only one checkpoint, that's about the ceiling. We can get response rates up to 50% if we block 2 or more checkpoints. The problem with that is with each checkpoint blocked, the toxicity goes up to the patient. And that really is the limiting factor in all of this. The more immune function you enable through blockade, the more toxicity, the more autoimmunity the patient receives. And that is literally the biggest challenge, because the benefits and the risks start to unbalance. So you can see in this graph, these are response rates at 4 years. That's, we're very happy. This is where there were no responses. 38% stability at 4 years, and then down to 34 at 5 years. That's exciting, but that is the ceiling. So there is 65% of people, the rest of that ceiling needs something done. So if you look, and these are the responsive diseases. So melanoma and kidney cancer and those kind of things, they would be considered hot cancers. They're responsive. The immune system sees them and it is enabled in and of itself. And then we can enable it further by using these checkpoint inhibitors. But a lot of the other cancers, as you can see, lung cancer, colorectal cancer, a lot of the head and neck cancers, they're falling under 20% response rate. And in the vernacular, in the jargon world, we call them cold cancers. And so the challenge has been to this point, giving more checkpoint inhibitors might enable a weak immune response rate, but it also enables that autoimmunity that we're trying to prevent. So what was the solution? And we believe we've come up with the solution.
And of course, while we're starting in melanoma, we're targeting specifically the cold cancers down here, colorectal, breast, bladder, potentially, where we think we can make the most difference.
Well, exactly. So melanoma, just a great point, David. So that we start in places like melanoma because A, they're readily accessible, easily measured, and there's an abundance of it in the clinics that we are currently working in. Cold cancers like colon cancer, which 90% are cold. You will have heard about colon cancer being treated with immunotherapy. It's a very specific subset of colon cancer, but for the 90% majority, they're not responsive. And so this is where our current research is focusing on bringing those cold cancers into the immune therapy arena and making them hot.
Really moving them up this curve, if you will, I guess.
Exactly.
Yeah, okay. And the new data we have today to discuss that was released last week is in the colorectal realm, so we'll get into that in a moment. So, Carmen, won't you take us through what the targeted hyperthermia therapy here is?
So, targeted hyperthermia, what this essentially is we're taking the gold nanoparticles, we're injecting them directly into the tumors, and in doing so, this is a preclinical mouse model, of course, the standard of care for all immunotherapy. We grow a tumor on the animal, then we fill the tumor with gold nanoparticles. We don't give it systemically at this point because we just want to concentrate the gold in the tumor and not be concerned about distribution throughout the animal. That's a safety feature, and it actually helps us to understand what we're seeing. Then when the tumor is infiltrated with the gold, we pass near infrared light, laser energy, through that tumor. The gold that's within the tumor absorbs the energy. The laser is about 860 nanometers. The gold particles are about 60 nanometers by 15. So this combination of things like a tuning fork, when you pass the energy through the gold nanoparticles, they vibrate. The vibrational energy creates heat. So it turns the light energy into heat. And we monitor the heat so that the heat never goes above 48 degrees. That's like high fever heat. Now, extremely high fever heat, I'll give you that, but that is for cells of the body. Your cells can tolerate 45 degrees quite comfortably. Not you physically not comfortably, but the cells no harm comes to them. But in cancer cells, they're not very well stressed. And so when we heat cancer cells, little proteins in the cancer cells called heat shock proteins are unreleased. And in releasing heat shock protein, we activate an immune response, an innate immune response. That is the immune response that responds to stress in general, and this being a stress. And so this is we're capitalizing, exploiting, if you will, a function of nature, which is to heat tumors specifically. And in the old days, in my practice, we would heat a whole body to do the same thing. Now we've got this very elegant, targetable hyperthermic therapy that we can put directly into the tumor. And in doing so, that causes the tumor cells to die. When the cells die, your innate system, the same system that heals a wound when you get cut or after you have surgery, that system comes in and causes a healing effect. It chews up the dead and dying cells. And in doing so, it constantly shows those dying things to the immune system so that your immune system knows if this is new, if it's foreign, if it's due to an infection. And if the immune system hasn't seen this protein, which is essentially what cells are made out of, then it will continue to send out fighters to eat that protein until it's gone, until it's cleared. And this is the innate immune response that we're hoping for. And thankfully, I can say we see it in every cancer that we've treated. So this laser energy is the elegance of it is, again, we put the gold into the tumor and then we heat the gold, passing the laser through it almost on a cyclical format, like a harmonic format, so that the temperature goes up and down between 42 and 48. So at about 45 degrees, we're comfortable that we're not killing other cells, and so we don't see any other cell death. The tumors, all the cells, the tumor cells die, all the cells around it are maintained. The heat is in for 5 minutes, it stopped, the stress goes away, and the natural process of cell death and immunogenic cell death, meaning that the cell that's dying is generating an immune response, carries on. And we've proven this in our experiments.
And that's a critical point you make there, that the gold nanorods, why they are the ideal and the superior nanoparticle for this application is that they have the most efficient conversion of non-thermal energy into heat. And so if we just turn on the laser and let it go, that heat will continue to rise up to the point where all cells are being damaged. And so this harmonic effect is keep it in the band between 44 and 48 in order to give it just what's needed to kill off the cancer while preserving the healthy cells.
Exactly. That's critical to the whole process.
Okay. So what's new? Let's get into this colorectal data. And Carmen, do you want to take us through?
This is again an industry preclinical model. This is a cold colorectal cancer, not the kind that we currently use immunotherapy for. And what we've done is we've taken this cancer, we've grown it in the flank of the host animal. And then you see this first line here. This is if you put the tumor cells into the flank and just nurture the most, the tumors will grow, and they'll grow pretty much unbridled. Slow, steady growth. You can see the way the curve goes. There's kind of an uptick. This is the old, think about it this way. If you have 2 pennies and then you multiply them, 2 becomes 4, becomes 8, becomes 16, and so on. So there's an exponential curve of growth. That's the natural curve of cancer growth. When we treat with PD-1 inhibitor, which is what that gray curve is, the PD-1 inhibitor, again, the checkpoint inhibitor that blocks that, if there's an immune reaction in that tumor, then the PD-1 inhibitor should enable that immune reaction and diminish the growth of the tumor. To what extent is depending on a whole bunch of factors. But what we see here is there's essentially no effect from treating these tumors with direct injection with a PD-1 inhibitor or systemic injection. We've done it both ways. Next. So this is, if you repeat the experiment, and we've done it a couple of times now, so we're seeing 2 cohorts, meaning we've got a group of mice, we do it too, we repeat the experiment with another group of mice. Twice we do this, and we see that if we first treat the tumor with targeted hyperthermic therapy, in other words, if we turn on that innate immune system so that the tumors are now being presented to the immune system, the protein in the tumors specifically, and the immune system now sees that as an immune target, then the checkpoint inhibitor, the PD-1 inhibitor, again, following the logic that we've talked about, will be now effective in an area where it wasn't effective before. And that's exactly what we see. Those lines are essentially flattened. This is so when you look at these, when you look at the tumor volume, you have an initial growth, of course. You have to wait until the tumor has volume to inject it. And then when you have an injection point, no more growth. So we arrest it. Now, what is some of that, when you look under the microscope, some of that volume is actually structure to the tumor, the supportive scaffold, and tumor cells within it are dead. But as an indicator, what we're seeing is we've arrested growth. Those are essentially flat lines. You see the blue line where things, now, why do the lines stop? That's a function of the experiments. When we write the protocols to the animal ethics people, we have a finite amount of time we're allowed to keep those animals alive for humane reasons, obviously. And so we're forced to terminate the animals and sacrifice the animals so that we prevent the suffering. So these are finite numbers. Essentially, our most recent protocol got us to 26 days. And if you the blue line again, we were really quite surprised. Not only did the tumors respond, the immune system completely cleared them. If you had an ulcer on your hand, envision the ulcer completely healing and the skin returning to normal. This skin was essentially normalized from the tumor. So that was above and beyond. Now we're excited about that, but that's, if you think about it, everything I'm telling you, the immune system should get you to that point eventually.
So now we wanted to break down, because these lines represent the average for the, it's the aggregated data for eight mice effectively in each one. What does it look like when we break them out? So we wanted to break that out for you a little bit and show, in this case, Carmen, this is just the cohort treated with immunotherapy.
Right. And so these lines, so we broke this down for you to see that these are mice that are these are tumors grown and then treated with PD-1 inhibitors. And what you see on every one of those curves is that the curves turn upward. There's lots of factors that go into tumor growth. When we show you an average, we're showing you a line that's an average of 8 lines. And so some tumors grow slower than others, some tumors grow faster than others. What we can tell you from these curves is that the treatment with the PD-1 inhibitor had no effect on the trajectory of those lines. They continue to go on in whatever growth rate that they had started with, largely because there's no effective immunity happening in those tumors. Now, we've also disassembled the ones that we treated with the targeted hyperthermia and the gold. And if you look at that slide, you see these lines. And so this is every mouse, that's 100% of animals treated have responded to this therapy. That in a laboratory setting is in and of itself astounding. So this is reassuring. This is from this data, all you can say right now is that targeted hyperthermia activates immunity, and without it, there is no effective immunity.
Yeah. And it's clear that in each case, the mice treated with THT did better than each instance of mice that had the best-in-class current standard of care immunotherapy. Right. That to us is compelling.
Yeah, and that's right. And that's exactly it. So again, this is essentially addressing current standard of care and showing that we can make a difference where it doesn't work.
Mm-hmm. Now, other measures, this is, of course, tumor volume that we're looking at, but we also often, you will have seen past press releases of our studies talk about gene expression analysis. Yes. And so we wanted to spend a little bit more time to really explore and explain what that means, what we're looking at.
Yeah. So this slide, this is an important, now I've been saying over and over, this is an innate immune activator. And what does that mean really? Well, when we activate the targeted hyperthermia by passing the laser light through the tumor, so one might say, well, it's just dying. So that's just cells dying. And so the tumor shrinking, so what? Because, that's what does that got to do with anything? Well, what we wanted to show was that cell death, it's the name in immunotherapy is called immunogenic cell death. As the cell is dying, it's dying in such a way that the immune system takes its components and represents it to the immune system so that the immune system becomes aware of it in a different way. And so if you look at these things, we look at a panel of genes. So these are essentially the genes or biomarkers, if you will, of innate immunity. These are interferons, IFN-A1, that's interferon alpha-1, alpha-4. The interleukins that I use in the clinic, interleukin 12, 18, 1, etc. The TLR down the bottom. These are indicators of innate immune cells. So macrophage and the neutrophils that may not mean anything to you specifically, but what this is when you get an infection, these are the cells that will be turned on to fight the infection, and they will stay active until the infection is cleared. All the blue, if you look at the scale on the right, essentially all of the blue or shades of blue essentially relate to fail or no expression. So a dark blue is zero expression. And as you move up, if you see moving towards the green and then the yellow spectrum, these same genes are now being turned on. So what this control panel is the panel that received just laser light with no gold in the mix. And then if we treat at the tumor with our THT, the next panel will show you that we've activated so many of these cells. And so down here, the toll activators, the yellows down at the TLR 7, 8, and 9, that's essentially that's a current target of immunotherapy in the clinical world. And we activate it simply by stressing. If you look at the other, there's lots of greens and yellows. So the CCL7 and CXCL10, those are receptors that are actually calling immune cells in to the tumor that weren't active on the other side. So these are all activators. Interleukin-1, and you see the yellow in the middle of it. Interleukin-1 is the number one. So it's calling in a whole bunch of other innate cells. This is just activating strong local tumor immunity that we know now translates to active immunity that can be further enabled by turning off the checkpoints, because the natural response to immunity is to check itself. And so that's where the further therapy comes from.
So just to be clear, each column represents a different mouse here, and then the various biomarkers for each. This obviously on the left being the control group, and this being the THT group. So Carmen, would it be fair to say that, let's say this second mouse treated right here with THT lit up, that presumably if we looked at the volume of that tumor, that it would have seen the most shrinkage?
Yeah, no, you want to believe that it saw the most immunity. How that translated in terms of tumor volume, there's other factors to go into growth of tumors. Sometimes tumors look like they're growing because they're just getting flooded with immune cells. And so we call it pseudogrowth in the jargon in the clinics. So when we treat a tumor, the tumor looks like it's getting bigger. We say, oh my goodness, it failed. Then if you look at that tumor under the microscope, it's mostly immune cells just flooding the field because it's so strong. So we don't want to be too linear in the relationship, but what we want to say is, oh my goodness, that's Christmas. All the lights are activated, and so we're seeing good, strong immunity.
And these are stronger or more important metrics for us than just shrinking in and of itself, because this is fundamentally what's going on in the body.
Absolutely. Within this kind of data are the keys. This is where we can further tweak the immune response. So this brings us the enablers to know if we take our THT plus a PD-1 inhibitor and we link them together, can we drive it? If we take that and use an IL-2 or whatever. So this is where the biology is really sitting in that information.
Excellent. Okay, let's move on here. Thank you for that. Now, safety studies, as we've spoken to in the past, in our pre-submission meeting with the FDA, we ran through our program of safety studies that we intended to undertake. And we have been doing just that, and we have been very pleased with everything we've received to date. We have a major safety study underway right now that we expect to get reported back to us in a couple of weeks. And so I want to provide a little bit more. First of all, sorry, the first one is on the laser device itself. There's a degree of course, our therapy is based on a two-device system, our gold nanorods and our laser. There needs to be a certain degree of work of validation and verification of those devices before we submit to a regulator. We've been working with a top-tier medical device maker here in Canada to do the verification work on our laser. This is some of that data, and effectively what they're assessing for is the laser actually applying the energy that we claim it is? And so that's important, data and a certificate to have. And we've come out very pleased with that. It's plus or minus 3%, which is incredibly good, and gives us the confidence that what we're applying is what we say we're applying and is what we need. Now, on the tests the testing, the study that's going on toxicity, et cetera, currently. It's our first so-called GLP, Good Laboratory Practice study. What that says is frankly less about the standard of the laboratory and more the fact that these results, when they're undertaken, must be relayed to the FDA, regardless of what the results are. And they go straight from the third-party lab to the FDA. They do not stop go. They do not collect $200. What we're assessing here for are effects of what the maximum feasible dose is. And we really are, going to well above and beyond what could ever be anticipated for any particular physiology, just to really stress test it. And this is something the regulators need. Second of all, the histopathology, looking at where the gold nanorods go in 10 different types of tissue inside the body. And finally, where do they distribute and how do they clear the body, both in terms of the path and the timeline. So as we anticipate these results, what we want to see and what success is going to look like for us is that there's no indication of gross toxicity from the gold nanorods at any level, let alone, at the lowest, and which would be the standard for what we'd be applying in a human context, let alone the stress test. And of course, secondly, the indication of clearance dynamics, understanding that clearance, the half-life, and the path followed. Important information that is going to be a critical component to provide to ethics committees prior to first-in-human studies. Okay, so with that, where are we going? First-in-human early feasibility study. Let's talk about that a little bit. So first and foremost, we've kissed a few frogs along the way here, but we are extremely happy with where we have landed and where we are right now with that study, in that we have engaged an excellent clinic that specializes in running clinical studies and has done a great deal of work for the likes of Merck, Regeneron, and many others. It's what they specialize in. Really pleased with that development. Jurisdictionally, for us, the factors were we needed a jurisdiction where there was a high incidence of melanoma, number one. Critically, that immunotherapy is part of the standard of care. We need a relatively large population just so that we get the throughput and are able to do this early feasibility study as quickly as possible. We get enrollment quickly. And of course, a favorable regulatory environment and one that is favored by the FDA. All of those boxes have been checked. So very pleased with where we are right now. Status-wise, our study has been filed with the local ethics committee and is pending. Now, we are still pushing like mad to get it started in the second quarter and expect that to happen. But of course, look, there are a lot of variables at play here, and over which we have very little control when dealing with these matters. But still confident that that is going to happen, and every indication is that that's going to happen right now. So let's take a walk through then what this study looks like. First and foremost, primary objective is safety. It always has to be for an early feasibility study. It's going to be secondarily, of course, the efficacy. And efficacy from the perspective of reduction in size of the tumor. And this goes back to our prior conversation with Carmen and how that is one indication, but the exploratory objective here is going to be understanding the immune cell infiltration and what the dynamics are within the immune system, which, as we talked about, is in fact more important. So all three very, very critical components. Carmen, do you want to talk about the protocol?
So essentially, yeah, the protocol, patients will be selected by a primary investigator. That's an oncologist, medical or surgical, currently treating the patient. And so the patient will have certain criteria. They'll have numbers of accessible melanoma tumors. And by the treater's assessment, the tumors will have been stable or not responded. So non-responding tumors under current standard of care will be selected. The patients will be otherwise healthy. And then what we'll do is once they're selected and enrolled and given consent, we will measure the tumors, measure a whole bunch of metrics, take some blood from the patient to know that they're healthy, and we know their kind of standard parameters of health, liver function, kidney function, etc. And then we'll do a biopsy of the tumor. And the biopsy of the tumor, of course, is to understand the current status of the immune system as it relates to that tumor. A small 2 millimeter punch biopsy will be taken. Then on the first day, we'll apply the THT. So we will inject a volume of gold nanoparticles directly into the tumor in multiple sites, and the volume will be calculated. We have a volume calculation curve based on the number of particles it would take to distribute throughout a tumor of a given size. Maximum tumor size will be 2 centimeters. Largely for it's a feasibility study, and so we have to make it manageable from a treater's perspective and a patient's perspective. And then we will apply the heat, pass the laser through the tumor, achieving a temperature between 42 and 48 degrees for 5 minutes. That can take 10 minutes in total if you think about warming up the tumor and then maintaining the heat. That's the extent of the tumor. Patients at the same time will be given quality of life questionnaires. They'll be assessed for pre-treatment symptoms and then post-treatment symptoms to see if there's anything relatable to the treatment. And they'll be monitored closely, essentially through clinical follow-up. 8 days later, we'll repeat the process without the biopsy. It's too early to do a biopsy, we feel, and these are patients, so we respect the nature of these invasive type tests. At the end of so 2 treatments of targeted hyperthermia will be applied. The patient will continue on their immunotherapy trajectory, whatever that is. Canada is literally the only country in the world, and especially Nova Scotia, that uses intralesional therapy on a regular basis. Most people will be on some form of systemic immunotherapy. And then at the end of the study, at day 30, we will do a final biopsy to see what the effect was over the course of 4 weeks under the influence of the targeted hyperthermia and in the context of their ongoing care. We'll also measure the volume of the tumor, expecting that tumors will shrink eventually, regardless of how much cellular infiltrate it is. But what we want to look at is not only tumor shrinkage. We don't expect tumors to clear. That would be a wonderful bonus, but we expect tumors to shrink. So any reduction from the initial tumor size and any improvement in the immune profile of the tumor will be, in the context of a well-tolerated treatment to the patients, will be the marks of success in this study. That's what we'll report on. From there, and as I said back when I showed you that slide about the gene study, from there, the information we get from this will help formulate the next protocol, which we are currently working on, and we'll want to put this information into that plan.
Yeah, obviously a lot of learnings that will come out of this. But suffice to say that we're extremely pleased with where we are, very pleased with the partner we have selected, and just roaring to get going here. The key question for us, and this flicks to the next stage here, which is really around timeline, is how quickly will enrollment take place? How long will the ethics committee take to approve it? Other sign-offs that are required, how long will that take? And then how quickly can we get enrollment? But in any event, when we look down the calendar ahead, looking for this, first and foremost, we have biocompatibility feasibility study results coming up. We've spoken to that. The verification work on the laser, and we're developing a second laser for redundancy purposes. Thirdly, the actual dosing of our first patient in that study. You see life science companies, any clinical trial, it's what is announced is first dosing. It's not contracting with a party. It's not starting a trial. It's, nothing is real until you're dosing. And so that is going to be a critical milestone for the company right there, first dosing. And then hoping to get initial readouts from that study in Q3. Again, it's subject to enrollment. We will, depending on how things go, we'll see how quickly we're able to put out some results based on how much enrollment we get. And then Carmen alluded to the idea that we'd be taking learnings from this study to apply it to our pilot study. And the pilot study is one, both of these studies have now been filed on clinicaltrials.gov, which is a first step towards it. You'll notice the timelines indicated, if you look, are really stretched out, but that's to give us scope, and that's a standard protocol. It also we'll want to have a 1-year timeline to do follow-up on these patients as well. Now, something we haven't talked a lot about are some of these other developments we have in the works, but we have a tri-conjugate. These are future products that are being developed that really show that our gold nanorods and the biocompatible nature of them makes them this ideal system to use in vivo. And so we're looking to leverage it and make it a platform technology, which is, in this case, work is ongoing in the background on these, not much by way of resource being put into it, but a little bit, because of course the full thrust and focus of the company is on getting these first-in-human results. Nonetheless, we'll talk a little bit more about this as we talk about future IP development advancements. The company's only as strong as its IP. We've got great core patents, and there's more we wanted to talk about a little bit now. There are a bunch of questions coming through, and we will get to the questions, I promise that. How many patients is ideal for your initial trials? I'm going to go back to that. The early feasibility study 10 is what we want for this initial study. Fewer would suffice, but that is the target. Pilot study would probably be 30 to 40, and then pivotal trial would likely be in excess of 80, to be determined in due course. But obviously ramp it up as we have results that show our comfort level in there. Patent-wise, we have our 3 core patents that are held by the company. But importantly, we've been making provisional filings of new ones here, both on the light device, but in these 2 in particular, these combination therapies that use the properties of the gold nanorods to take things to the next step. And if you follow other life science companies, you know that all the rage in the last 5 years or so have been on ADCs, antibody drug conjugates, the ability to use antibodies in the system to attach them to a particular drug, which then serves as a cruise missile, if you will, within the body to go directly to a targeted site. But Carmen, do you want to provide a little bit? We're not able to speak that much to these concepts at this stage for a variety of sensitivity reasons, but do you want to speak to them?
Yeah, as David indicated, the objective here is to target distant disease. And so we know we have laboratory evidence and good sound biological evidence that if we can get the gold nanoparticle to the tumor and activate it, we can generate immunity. So a tri-conjugate means we're going to take gold, we're going to combine it with a homing device, and when people have PET scans, the PET scan is kind of a homing device. It's targeting tissue that's chewing up sugar quickly. If somebody has prostate cancer, though, they put a prostate-specific homing device on the imaging. So that radio tracer that they inject into the vein goes specifically to where cancer metastases are. And so you can imagine if you had something attached to that imaging process, when it gets to the cancer, you can flick the switch and activate some immunity to the cancer. That's the conjugates we're working on. And there's a number of them. We're very excited about it. We've engaged another lab at Dalhousie. We're funding some students to work with this, and we're putting IP in on this. And this is honestly the platform technology. The gold has, like an antibody drug conjugate, where the gold is actually carrying a drug to the tumor, and in and of itself is a device to treat the tumor. So it's the marriage of every world. It's superb.
Yeah, and you can think of the next several iterations and evolutions of targeted hyperthermia therapy, which will really, once we prove it as working in humans, to be able to really enhance it and improve it through these leading edge methodologies will make an enormous difference for the company. Really excited about this. I'm going to skip back to, there's a question here, Carmen, on if the early feasibility patients will already be on immunotherapy drugs, how do you differentiate the effect of THT from the drugs?
Perfect. That's exactly the question. So the people that are on immunotherapy drugs who are not responding, we know are not responding because the immune relationship in the tumor is not strong, whatever it is. So that's what the first biopsy is about, is to understand what does that immunity look like. And often what we see when we biopsy patients in our world today is we see some immune cells, but the cells aren't activated. The T cells, the ones that are fighting the cancer, don't have PD-1 receptors, which means they're there, but they're asleep. And so what we'll show is that once we've treated the tumors with the targeted hyperthermia, we'll have activated fresh immunity so that the immune process that's being targeted will be enabled. And so that's great they're on an immune therapy, because what we're showing is the immune therapy wasn't working, switch on, now it's working. And so, and the switch, of course, is activating that fresh new immunity that we know is in the tumor. We just got to get it exposed. Yeah. And so this is, it's actually without that, we wouldn't be able to tell you that we had it. We were the big factor.
Yeah, we're really pleased with the design of the study, where it's going to be taking patients who are on the best immunotherapies in the world and they're not working. We then apply our THT, and if suddenly they work, it's crystal clear what the difference maker is.
Yeah. So I'm just going to say, Dave, if I could, there was another question that flashed up, and I think it might be a very bright, informed question. And that is, that data that I showed you with the gene panel, was there immunotherapy involved? And the answer is no. That is, we want to specifically show that this is just hyperthermia. No hyperthermia on the left, hyperthermia on the right. And we controlled it very carefully. We used laser because we know that laser in and of itself has some interesting effects in tissue. Laser only on the left, laser and gold on the right. And so this is without immunotherapy. So this is completely innate immunity. This is generated by the tumor being presented to the immune system in a better way, if you will.
Another question here, assuming EFS success with hot melanoma, will the next pilot study be done with cold tumors?
Yeah, so another great question. So we're working hard in colorectal cancer right now, because that is absolutely a cold tumor, and not just colon, but rectum. And the rectum is a super challenging area for those of you that know people that have had rectal cancer. It's treated now with chemotherapy and X-ray therapy. And the X-ray therapy is treated to the rectum. It only has effect in the rectum. It has no benefit to the person beyond the rectum. And over a number of years, because it's radiation that's toxic and causes all kinds of dysfunction, these people, the quality of life is diminished over the long haul. What we hope is that we can replace the radiotherapy with the targeted hyperthermic therapy to cause the local tumor shrink, and in doing so, generate that immunity that wasn't generated in the natural process. So yeah, that's our cold tumor is definitely colorectal cancer will be our next target.
Yeah. Excellent. So we've been through this work here. The other advantages we have, of course, are time advantage and a number of trade secrets, if you will, in terms of how to, because we are on the leading edge of this, how to apply these concepts. Let me just go to a couple other questions here too, in terms of, are there any drug companies currently monitoring your progress? Well, we know that lots of people are reading the study that was published in Frontiers of Immunology in January. It's in the top 5%, I think, of its relevant group for readership. And of course, Carmen recently presented it at the Canadian Melanoma Conference. A lot of drug companies there. Look, drug companies get their information from two sources: conferences and published reports. So we'd expect that's the case. We were approached by a few. Those conversations, would continue. But there's lots more work to be done here. And in fact, it's from the company's perspective, way too early to be having those conversations, because we have so many things that we're excited about pulling together in the next few quarters. So pipeline-wise, starting with melanoma, this goes to that conversation around the cold tumors versus hot tumors, but we hope to follow that up with colorectal next. It's a big problem. You saw early on in this deck that response rates on immunotherapy are very low on colorectal. It's a cold tumor, and perhaps we can have the biggest delta there in terms of the impact, the efficacy with our targeted hyperthermia therapy. Likewise, breast cancer. Carmen, do you want to speak to these other ones as well?
Yeah, so as I said, rectal cancer is the next obvious. Like melanoma, it's readily accessible, but breast cancer, we actually have a study with interleukin-2, ours the only in North America, in triple negative breast cancer. Many of you will know that triple negative breast cancer is actually immunogenic, and the response rates are actually quite good. So, like melanoma, we're going to be targeting that as a way to enhance the response with that 50 or so percent of people that don't respond to the current immunotherapies. And it's otherwise triple negative breast cancer, which is, as everybody that knows something about breast cancer knows, is the worst of them. But that will move into the immunogenically cold cancers once. So breast cancer is a two-step process, but we're excited about that, and we're very excited that we're on the leading edge in terms of intralesional therapy in breast already. Glioblastoma, very excited about this glioblastoma. Some of you will know that there's actually a microtherapy using heat that's being used in some clinics now in glioblastoma. We know that the brain can be treated with an immune therapy and can be immunogenic. And we were delighted to learn that we can actually target the brain using another specific test that I really shouldn't disclose right now. But yeah, so glioblastoma, the brain tumors, we know we're on there. Bladder cancer, we're working with a colleague in urology. Bladder cancer, some of you will know, is already treated with an immune therapy called BCG. It's horribly toxic and incomplete always. It's a lifelong exposure to this that leads to a lot of the complications. And we're certain that bladder cancer, given that we know how it works with immunotherapy, this is the better therapy. So we're excited to go there. And then cervical cancer and vaginal cancer, or those mucosal cancers, and you can include rectal or anal canal cancers. Those are cancers that tend to be immunogenic. And the immunology around those areas is very challenging, but we know that the hyperthermia has this ability to reinvigorate immunity. So these are accessible. So those are kind of the short next step kind of cancers that we know we can get to with very little modification in the technology that we're applying currently to melanoma.
So time is running on here. Of course, the other, the tri-conjugate and diagnostics obviously added to our pipeline as well in the early stages. Just to wrap things up here and turn it over to some more questions. Obviously, we think we've got a very powerful product here on our hands. We've got a unique properties from our platform technology, and we have compelling data from our preclinical efficacy studies, and we are on the cusp of a first-in-human study that we think will be a major catalyst for the company, with a strong and growing patent portfolio, a great team, and a connected board. We think we're well positioned, and we are excited. So at this point, I'd turn it over and ask if there are any other questions from the group. Let me look through. There are a bunch in here where we may have missed. In particular, where did that one go? Yeah, now's the time you can add more in. Questions here about asking us to talk about pursuing either a 501(k) or a PMA approval. These are the different paths to approval in the FDA regime that are important. And there's a risk assessment work that happens for that. It's early to determine exactly what path that will be. Suffice to say, the safety study data coming out in the next few weeks will be critical to determining that, as it will determine, it'll dictate, give an indication as to how long gold nanorods last in the body before they're all flushed out. And we'll be looking forward to our next conversation with the FDA to help assess that path. Regardless, pushing forward both in Canada, given the clinical trial, the next pilot study will be done in Canada, and looking at both jurisdictions, and therefore talking to both sets of regulators. In the human tests, will you also be evaluating non-treated tumors, Carmen?
Yeah, so the short answer is no, not in the first 10. And the reason is we have to really focus, the whole study is focused on the toxicity, safety, and somewhat efficacy in those tumors. We wouldn't, there's, in terms of the studies, we're only allowed to do so much with the patients. And so we're focusing on that intralesional tumor only. The next study will go to a broader reach.
That's it. Excellent. Okay, well, unless there are any other questions now, please feel free to reach out anytime with further questions. We'll be keeping you up to date. I think, lastly, I think you've seen, if you're following the story closely, there's been a lot more engagement with the capital markets, and you'll see lots more coming from us, lots of milestones coming up, and we could not be more excited. Everything we're seeing so far is looking great. Thank you, everyone. Thank you, Carmen. We'll do this again soon. Cheers, everyone.
Yeah.